Hello students, hope you all are fine. Stay safe, stay healthy. Here is Dr. Hira Bashir, MPhil Pharmaceutical Chemistry. And today we will discuss about bacteria. As we know, bacteria is a microorganism. We have discussed earlier that it is a prokaryotic species which is very small in size, and we use microscope to see this microorganism. To continue our discussion, we will discuss here the growth and reproduction of bacteria. Growth and reproduction of bacteria. This means how bacteria grow and reproduce, how bacteria increase its number. This is basically division of one bacterium into two daughter cells by the process of binary fission. Bi stands for two and fission means breakdown of a single cell of bacteria into two. The resulting daughter cells are identical to the mother cell. Hence, local doubling of bacterial population occurs. Local doubling means doubling of the number, increase in number takes place. This is the basic binary fission process, in which you can see here a single cell of bacterium in which there is a genetic makeup present in the center, basically a DNA molecule. This DNA molecule transmit and share its material, divide into two, and then these two cells, nucleus cells, separate out, and each nucleus is present in each daughter cell. Single cell share the material, stretches towards both sides, and the single cell is divided into two different daughter cells, exactly similar. You can see here DNA replication. Replica means making an exact new copy. Then cell elongate, means cell increases in size, the second step. Then formation of septum, means the formation of layer. This septum is formed between these two cells, and the cells separate out. You can see here cell separation. Then each daughter cell receives one copy of chromosome. Both the daughter cells have the same copies as parent cell. This one is a parent cell and these two are the daughter cells. This is the basic process of reproduction in bacteria. Then bacterial growth. How bacteria grows? This is the process of reproduction that the bacteria reproduce by binary fission. Then how it grows means how its number increases. Increase in number of bacteria is called bacterial growth. Important environmental factors affecting bacterial growth are temperature, nutrients, means food, pH, mean acidic environment or basic environment. Alright, acidic environment or basic environment, and the concentration of ions, mean positive ions or negative ions disrupt the process of bacterial growth. 4 distinct phases of bacterial growth. Basically, it involves 4 different steps. The first one is the lag phase, in which bacteria basically prepare themselves for division. Bacteria prepare itself for division. Then log phase. Bacteria grow and multiply very rapidly. We can also say exponential phase. Bacteria exponentially rises in number. Bacteria increases in number very rapidly. Then the third one is the stationary phase. means static phase. The number of bacteria grows equal to the number of bacteria's death, means growth rate equals to the death rate. Then there is the decline phase. The death rate is more than the multiplication or growth rate. More bacteria dies as compared to the bacteria grows. In graph you can see here, the first one is the lag phase, preparation time for the bacteria to grow. Then exponential phase, bacteria increases in number. Then stationary phase, static phase. You can see here, if 2 bacteria grows, 2 bacteria dies. If 40 bacteria grows, then 40 bacteria dies. It's a static phase. Both rates are equal. Basically we can say here, death rate is equal to the growth rate. Then suddenly there is a phase of rapid death, decline phase. If 4 bacteria are formed, then 6 are dying. The growth rate is less in decline phase and death rate is higher. This is basically the bacterial growth. Alright, then the introduction of bacteria ends up here. Now we will start a new topic related to bacteria, but somewhat different, as now we grow these bacteria, and to grow these bacteria we require a media. We required any source by using which bacteria grows. Bacteria increases in number artificially, basically in laboratory, how we prepare bacteria, and that bacteria are termed as culture. If we are preparing bacteria by using nutrients, then the bacteria is called culture and the nutrients are called as media. Alright, you can see here different types of culture medias. Some are present in bottles, some are in test tubes, some are in petri dishes. These are basically petri dishes, these are bottles, then these are test tubes. We prepare different medias depending upon the nature of bacteria we have to grow. Different types of medias are grown. Now, what is culture? It is defined as the microorganisms, as we are discussing bacteria, so we will say the bacteria cultivated for the purpose of study in laboratory. If we say it overall, then we will say microorganisms cultivated, maybe a bacteria, maybe a fungus, for the purpose of study in laboratory. Only for the purpose of study in laboratory. Alright, because we are preparing artificially by providing artificial nutrition. This one is culture. Basically the culture means the living system which we grow is called culture. And how it is grown? Obviously by taking nutrition, which is called as media. The combination of ingredients used for the growth of microorganisms. Bacteria and fungi can only grow or cultivated by using culture media. Viruses cannot be grown. Only bacteria and fungi can be grown. Viruses do not have the metabolic machinery. Viruses do not utilize these ingredients. They do not have any machinery to metabolize glucose, to metabolize lipids, to metabolize proteins. That's why they cannot be artificially grown. But bacteria and fungi can easily be grown in artificial media. So if you ask about why viruses do not grow artificially, then you will say they do not have a biosynthetic machinery, basically metabolic machinery for growth. They cannot utilize these ingredients, but bacteria and fungi can easily utilize these ingredients. They have the metabolic machinery because they are autotrophic also. They can prepare their own food, but here we give them nutrition to grow them in the laboratory for the purpose of study. Then what is inoculum? It is a microorganism that are introduced to the media to initiate the growth. The microorganism, for example, we have studied about different types of microorganism. Take an example of Salmonella typhi, a typhoid bacteria. If we want to grow typhoid bacteria in laboratory, we obviously need certain number of bacteria of Salmonella typhi. So these bacteria utilize those nutrients and increase in number. That small amount of bacteria which initiate its growth is called inoculum. For example, if the number of bacteria grown are 50, then how these 50 are grown? We applied a sample of 2, 4, 7 or 10 microorganisms or bacteria which uses that media and grows up to 50 in number. So we required inoculum to start the growth of microorganism. You can see here is a petri dish, a glass dish in which we prepare a media providing specific nutrition. That we discuss later, how we prepare media, why we prepare media. Then this one is the loop. This is a loop in which a sample of bacteria was taken. You see here, petri dish plus media plus inoculum, means inoculum is placed in this loop and a pattern is drawn. If some irregular lines are drawn, how they are drawn we will discuss it later. Then we will draw a pattern with the help of that inoculum loop, and after certain time period of either 14 days, placing it in the oven for required time period at required temperature, you will see there is a growth of these bacteria. This whitish area shows it that the sample applied on this loop and then covered and placed in the oven for required time period. Then when after 14 days or 7 days it is opened, we will see the growth here. The pattern we drawn there, where inoculum is applied, the growth is seen. The number was 2 or 10 or 50, but in all the lines we see the growth. How they grow? By using this culture media, this media we prepared. Alright, let's move toward the other topic, the classification of media. Classification based on physical state, like physically seeing something and observing either it is solid, either it is semi-solid, either it is liquid. That is basically a physical state: liquid, solid and semi-solid. These types of media are basically discussed here. Solid medium, 1% agar. Now what is agar? It is commonly used as a solidifying agent. Alright, what is agar? It is a golden yellow granular powder prepared from seaweeds. You can see here seaweeds. Using this we process this material and then prepared this type of dried powder and used as a solidifying agent to prepare a media, not affected by the growth of the bacteria. Melt at 98 degree and sets at 42 degree. You can see here it melt at 98 degree, means the point near to the boiling of the water. Water boils at 100 degrees Celsius, and at 98 degrees Celsius the agar melts, and at 42 degree it sets in the form of solid. As we increases the quantity, the solid nature increases. As we decreases the quantity of agar, the nature become somewhat changes. Like if 1% is for solid, then we want to make it somewhat semi-solid, means half liquid or half some solid nature, gel type nature, then we decreases the quantity of agar. Alright. Colony morphology, you can see here pigmentation, hemolysis can be appreciated. All these methods are performed. Cell morphology is checked by using this type of media. Pigmentation is checked by using this type of media. Hemolysis is taken by this media. For example, nutrient agar, blood agar, available in petri dishes, and mostly it is available in petri dishes. Then liquid media, referred as broth. Bacteria grows uniformly, producing general turbidity. You can see here at the bottom it is clear and in the upper layer is turbid. This shows the presence of bacteria. Liquid media generally contain no agar. It contains no agar. It is liquid form, available in test tubes, bottles, flask, etc. Alright. Then solid, liquid, and now semi-solid. You can see here the percentage 0.2 to 0.5% agar. Gelatinous nature, jelly-like nature, gelatin-like, jelly-like. Such medias are soft and used in demonstrating bacteria motility and separating motile from non-motile strains. As we discussed earlier, different types of bacteria having locomotory organ are motile, and those which do not have a locomotory organ are non-motile strains. These are basically distinguished, means the presence of flagella is being distinguished by the presence by using semi-solid media. You can see here available in test tubes, available in petri dishes. Now, classification on the basis of chemical composition. The first one we have discussed is on the basis of physical state. This one is on the basis of chemical composition. Chemical composition. Natural, synthetic media and semi-synthetic media. The natural media is of further different types like nutrient agar media, beef broth media, enriched culture media, selective culture media, and differential culture media. These are different types of medias on the basis of chemical composition. And in chemical composition, we are discussing first of all natural type. Then natural culture media, basically this type of media basically contain the ingredients whose quantity is in which quantity of the component is unknown. We do not know the exact quantity. We take it randomly and like it subdivided as nutrient agar media, consists of water, peptones, beef extract, and agar. In its composition, beef extract 3 gram, we take it randomly, mineral source and carbohydrate source. Then peptones 5 gram as a protein and nitrogen source, sodium chloride 5 gram as an electrolyte, agar 15 gram as a solidifying agent, water 1000 ml or 1 liter having a pH of 7. How we prepare this media? By dissolving 28 gram per 1000 ml in distilled water. You can see here, if you add all these numbers, it comes out to be 28 gram and total volume is 1000 ml. We dissolve all these ingredients in 1000 ml of water by heating on autoclave at 121 degree Celsius. This is a method to prepare nutrient agar media. Then moving towards beef broth media, it is used for the cultivation of bacteria since the time of Pasteur and Koch in labs. At the time of Pasteur, beef broth media was used. Then selective media. Mannitol salt agar is used for the detection of Streptococcus species. Eosin methylene blue agar is used for E. coli species. Alright. Selective. It is selective type. It selects specific type of bacteria. Like if we have to test or grow Streptococci, then we will prepare selective media by using mannitol salt agar. If we want to grow E. coli, then we will use eosin methylene blue agar media. Differential culture media. It contains bile salts such as gram-positive bacteria cultivation and McConkey agar media. For the cultivation of gram-positive bacteria, differential culture media is used and basically bile salts are added in them. The media is called as McConkey agar media. Enriched culture media, as the name indicates, increased nutrients. The media used for specific species of bacteria. For example, nutrient agar plus blood. We prepared nutrient agar media in the first subdivision. In this media, if we add blood, then we have to cultivate Streptococcus species. And if chocolate agar used for Neisseria species, if we add chocolate, then it will make us chocolate agar media and for the species of Neisseria. Alright, to grow different specific type of species, we made specific type of culture media. Synthetic culture media. It is used for the cultivation of bacteria which have known composition and quantity of components. The quantity of component is known. We know the exact purpose, we know which component have to use, and glucose for the purpose of energy, ammonium phosphate for the purpose of nitrogen, potassium phosphate to provide phosphate, magnesium sulfate for enzyme formation, sodium chloride to maintain the internal environment of cytoplasm, as in ions they are used. Moving toward semi-synthetic. The media which have chemical composition half known. Some components are known and some components are unknown. Example includes nutrient agar, potatoes, dextrose, agar, PDA basically. Potatoes, dextrose, agar. Then some special medias are prepared. For example, enriched media, selective media, differential media, transport media, anaerobic media, and basal media. The basal media is basically a very simple media. It supports most of non-fastidious bacteria. Why it is prepared? Basically for non-fastidious bacteria which are able to grow without special nutritional supplements. Alright, or conditions applied to agar growth plates. It simply grows without any nutritional supplement. It is the simplest media. To prepare other media, we use this simple media and add nutrition that the name changes. Just like as if we have to prepare an sulfur ointment, we have to make a simple ointment, then we add sulfur into that ointment. Same as if we have to prepare any type of, for example, enriched media, we first take basal media, then add nutrition and make it enriched. Then enriched media is substances like blood, serum, eggs are added to simple media. Simple media means basal media. It is used to grow bacteria that are exacting in their nutrition needs. For example, blood agar, chocolate agar. Same example as we have discussed earlier. Then you can see here different color. Blood agar indicate red color and chocolate agar indicate chocolate color. Then selective media, the inhibitory substances are added to the solid media to inhibit commensal or contaminating bacteria, such as different inhibitory substances are added. For example, antibiotics, dyes, chemicals, pH changers, they are added to make the media selective. Then eosin-methylene blue, as we have discussed earlier, the example of selective media as for gram positive or gram negative differentiation, the dimethylene blue in the medium inhibit the growth of gram positive bacteria. Then differential media, as the name indicates, it differentiates the bacteria on the basis of color of the colony. On the basis of colony color, they are differentiated. Dyes and metabolic substrates are incorporated or added so that those bacteria utilize them appear as different colored. Dyes are added so different bacteria utilize different color and we separate them. Examples include McConkey agar, Sealed agar, TCBS, XLD agar, McConkey agar. It distinguishes between lactose fermenters and non-lactose fermenters. Lactose fermenters appears pink and non-lactose fermenters are colorless in the colonies. You can see here this one is a McConkey agar. We added dyes in it, and pink color indicate those lactose fermenters, lactic acid bacteria, and those which are non-lactose fermenters are appeared colorless. Then the second example was XLD, xylose lysine deoxycholate agar, used for recovery of Salmonella and Shigella species. You can see here for E. coli, yellow color colonies are formed, and for Salmonella species, black color colonies are formed. These colors are basically indicated in specific pharmacopoeias. You can confirm them, then write it here. There is a basic information about this. Then the third one is the cysteine lactose electrolyte deficient agar, CLED basically. For cultivation of pathogen from urine sample, inhibit swarming of Proteus species. Or you can see here CLED agar for E. coli, yellow color for E. coli, as we have known here, yellow color colonies, same as here. Then thiosulfate citrate bile salt sucrose agar, TCBS, highly selective for the isolation of Vibrio cholera, highly specific for Vibrio cholera and Vibrio parahemolyticus. These two species are specifically identified by this method. Using this media, we can selectively isolate these species. You can see here the color change, this yellow color and the other one green color. Thiosulfate, citrate, bile salt, sucrose, TCBS. Then the other media was transport media. The media used for transporting the samples. Delicate organisms may not survive the time taken for transporting the specimen without a transport media. Stuart media and buffered glycerol saline media is used. You can discuss it here as you can understand it by memorizing that if we have taken a sample of blood, we have to give a blood sample. You will see that that blood sample is taken to the laboratory or from laboratory to the specified area in a specific vial, and that vial contain a specific type of media in it, which is basically used for transporting that blood sample. Likewise, it is a media for transportation purpose only. It has no other nutritional value, just to transport the specimen without any disturbance or without destroying the sample organism. Basically, in glycerol saline buffer is taken, or Stuart media is basically used to transport. Then anaerobic media. These media are used to grow anaerobic organisms, those organisms which survive in the absence of oxygen. For example, Robertson cooked meat medium, thioglycolate broth medium. We can see here cooked meat medium for anaerobic bacteria. Here is a cooked meat medium. These are specific type of medias used to grow specific type of bacteria. Now, what are the techniques to isolate pure culture of bacteria? Like I told you earlier that we have taken a petri dish, in it we add culture media. Then we draw a sample, draw a basic pattern there. What is that pattern? How it is drawn? How it is drawn to isolate that culture? We'll discuss it now. Okay. Culture media is discussed thoroughly. All types of culture medias are discussed. Now we will discuss how that culture media is used to isolate pure culture of bacteria. Different methods are used. We will discuss only one or two here in detail, that how they are used to isolate. Isolate means separate, to separate that bacterial culture. The first one is streak plate method, the other one is pour plate method, then enriched culture technique, then serial dilution method, the single cell dilution technique, roll tube technique. Repeat it again: streak plate, pour plate, enriched culture, serial dilution method, single cell dilution method, roll tube method, etc. The first one is a streak plate technique. Now, what is meant by streak? Streak means to draw irregular lines basically. Streak is basically the irregular lines. It allows the bacteria to reproduce in a controlled environment. The bacteria are spread on the agar media at a certain temperature for a specific time period. It identifies the colonies of pure bacterial culture from mixed population. If we have a mixed population of bacteria, 3 to 4 different types of bacteria, then by using streak plate technique we can identify them very easily by drawing different patterns. Microbiologists use this streaking technique for diagnosis of infections. The inoculum, yani specimen of bacteria, is spread on the agar surface in the form of streak with the help of transfer loop. We will take a transfer loop having a sample, having inoculum of bacteria, then we make a pattern over there, and then on that pattern the bacteria will grow using the nutrition from the media. How? You can see here, we will take a transfer loop, we will draw this transfer loop contain the sample or specimen or inoculum, then we draw a, for example, first sample is taken, the loop is taken, dipped in the first sample, and this pattern is drawn as indicated 1. Then the second loop is taken, or this loop is taken, heated, make it bacteria free, then dipped in the second sample, second inoculum, then make another pattern, then again heat the loop, remove all the bacteria of second sample, then dipped in the third inoculum and draw the another pattern. Likewise draw fourth inoculum pattern. You will see here 4 different type of patterns were drawn, 1, 2, 3, and this one is 4. Okay, we can take single inoculum and draw 4 different types, or we can take 4 different types and draw one by one. This one is loop, this is a handle of the loop, this is a platinum wire, and the loop is made just about 2 to 3 millimeter in diameter. This one is petri dish. This petri dish contain media, and this yellow color indicate the presence of bacterial growth. This is inoculating loop. These are basic patterns drawn. You can see here, we put the loop here without putting it up. We will draw a pattern like this. Don't put up the loop. Continually draw this pattern and end up here. Then draw another pattern like this. Don't pull up the loop. Don't break the line. This is basically done on the media with a very light hand. You can see here we will heat it again and again. This is heat. First we add inoculum, then we heat the loop, then again taken the sample, then again draw this pattern, then we heat the loop, again draw this pattern, blue color pattern you can see here, then heat the loop again, add 4th sample and draw it here. You can see 4 different types of colonies with different color, appearance of colonies after incubation. We have to put it in at certain temperature for certain period of time. That one was a simple method to identify different colonies basically. If we don't want to isolate colonies, we can simply draw a single pattern like this continuous streak. You can see the arrow here. If we started from here, then we have to draw the pattern like this. Same in this manner. Then the other one, gradient streak. We have to draw the pattern first in the above, then in the left or right side. Then quadrant streak in the 4 different types of directions. First one, then second one, then third one, and then this one is 4th one. This quadrant streak drawn here. I told you 4 different type of colonies were identified. Just see here, this one is the right one, first colony. Stop it here, heat the loop, again take the sample and draw it here. Second colony, then heat the inoculum sample, heat the loop, take the third inoculum and draw it here. Likewise 4th one. 4 different colonies were identified. For single other colony, for single type of colony, draw the pattern like this. Then for radial symmetry, draw like this, and for quadrant symmetry, draw like this. Okay, we don't use these 2 techniques more often because they do not give as much as good result. These are the 4 basic techniques I have corrected. This method is not ensure 100% pure culture. Why it doesn't ensure 100% pure culture? Our air is almost full up with microorganisms. and we have the media which these microorganisms present in the air can be used. The microorganism present in air uses that media we have prepared for specific type of bacteria, but air microorganism use that media and start to grow on that culture media surface. Because the petri dish is open, we are drawing the pattern, we are exposing the upper surface of the media to the air. As all the process is done in a controlled environment like in a laminar flow hood, we use this technique, but somewhat air is contaminated and the presence of microorganisms are submitted to the media. They grow on the surface, therefore it is not 100% pure culture obtained. Then we perform pour plate technique method. You can see here it can ensure 100% pure culture development because very small surface area is exposed to the air. In this method you can see pour plate technique. The original sample, basically the inoculum of bacteria, is diluted to get isolated colonies. The sample is diluted with some media and the colonies are isolated, separated colonies are seen. The steps are basically take 3 test tubes. The test tube A incubated with 1 loop. We take a test tube, label them A, and 1 single loop full of original sample called as inoculum was taken and mixed. Then take 2 loops from this test tube A. From this test tube A, 2 loops are taken and added to the test tube B having media. Only simple media is present there, and we have added the media plus sample from test tube A. 2 loops full added in B. Then from B test tube take 3 loops, means 3 times take the sample and add it into the tube C. The tube C content is poured in the petri dish, allow it to solidify, incubate it for certain time period. Well separated colonies were seen. See this diagram. We have 3 different test tubes, label A, B and C. All 3 test tubes have culture media, simple culture media. Here is the inoculum. There is the inoculum of bacteria. From this inoculum, take 1 loop. Take 1 loop. Then 1 loop is taken, and this loop is of, you can see here, the small sample is taken from this inoculum and added in the test tube A. The test tube is then covered, sealed. Then take another test tube with the culture media. From this one having 1 loop of the inoculum sample. Now take a loop and 1 loop fill is added in this test tube, which is labeled as B. Alright. Then from this take 2 loops full and add it into the other test tube. You can see here the colonies formed are, if we separate all these, you can see different type of colonies with different gap between them. 1 loop full added in A, from A 2 loop full added in B, and from B 3 loops are taken one by one and added in the C test tube. All 3 test tubes are converted in petri dishes. The most important one we use for study is C, because it has isolated colonies. These are very closely present. These are also very closely present, but they are isolated ones. We can easily distinguish them, the other colonies present. Different type of colonies were seen here. Repeat this method then. Take 3 test tubes, these A, B and C, this one A, B and C. Then take test tube A with 1 loop of original sample and mixed. Take test tube A, add 1 loop full into it and mixed it. Then take 2 loops full of this sample from A and put it in B. You can see here, take 2 loops from here and put it in test tube B. Then take 3 loops full of this sample from B and put it in C. 3 loops full is taken from this and added in test tube C. Then this is put it into the petri dish and observed under microscope after certain time period, but visibly you can also see them visibly, and you can also perform staining with them. Here we only first of all prepare culture media, then that media is used to prepare culture. How the culture is prepared? These are these 2 basic methods, streaking and pour plate method, to grow the number of bacteria. You can see here, if original sample, all these have culture media, 1 ml of original sample is taken, diluted, you can see the colony here, difficult to distinguish. Then we taken 1 ml from this and added in this, you can see here slightly visible and separated colony, very slightly but different, difficult to distinguish. Then 1 ml is more taken and diluted, then you can see somewhat colony morphology is being distinguishable. Then further diluted, you can see here they are separated, and now they are fully separated after further dilution. Now we can easily distinguish it under microscope that which type of bacteria it is, either it is a cocci, either it is a bacilli, either it is what type of bacteria it is, we can easily identify it. Alright, these are basically different techniques to isolate these bacteria. We'll have a slight revision on this topic. First of all, we have studied about the bacterial growth. Bacterial growth and reproduction. We studied about the division of one bacteria into 2. Then the resulting daughter cell are identical to the mother cell. Doubling takes place. From the diagram, DNA replication, the first step, then the other step is the cell elongation, then third step is septum formation, fourth step is cell separation, and fifth one is the formation of daughter cells, which receives the exact copy of the chromosomes as that of the parent cell. Alright, then moving toward the growth, there are 4 different phases: lag phase, log phase, stationary phase, and death phase. Lag phase is a phase of rapid, is a phase in which bacteria rapidly prepare themselves for the process of division, for the process of fission. For the process of binary fission, bacteria prepare themselves. Then in the log phase, they multiply very rapidly. They increases in number, exponentially increases in number. Then in stationary phase, the rate of growth equal to the rate of death. The growth rate or bacterial multiplication rate is equal to the death rate. If 4 bacteria are formed, then 4 are dying. Then the decline phase, basically the death rate is more rapid than multiplication. We can say that death rate is more than the growth rate. More bacteria are dying as compared to the grown bacteria. In this graph, you can see as the time lapse, the lag phase is basically showing the preparation of the bacteria, Then the exponential phase, basically the log phase, increase in growth of bacteria. Then growth rate in death rate equalizes here in stationary phase, and then in the death phase or decline phase, phase of no growth, we can say a phase of the phase in which more death occur as compared to growth. Lag phase, log phase, stationary phase, and death phase. And culture media. What is culture? Basically, it is defined as the microorganisms grown for the purpose of study in lab. The microorganisms we are discussing here, bacteria, the bacteria grown for the purpose of study in the laboratory. Basically, we are preparing bacteria artificially. We are preparing them artificially for the purpose of study. How we prepare them? By using a media having proper nutrition, having proper ingredients needed for their growth. We provide this nutrition and they grow. They are cultivated basically. Combination of ingredients used for the growth of microorganism is called media. The bacteria and fungi can only be grown and cultivated by using culture media. But viruses cannot be grown because they do not have any metabolic machinery for the growth. Then inoculum, basically the original sample we take in the start of the process. Basically, to initiate the growth process, the original sample we take is called inoculum. Here is a list, a diagrammatical view. A glass dish, then media is added depending upon which type of microorganism we are cultivating. Then petri dish plus media, and then the loop is taken with inoculum and a pattern or streak is drawn. Different type of medias are taken as on the basis of physical state, either a solid, liquid or semi-solid. If it is solid, then it has 1% agar. If it is liquid, it contains no agar, and if it is semi-solid, it contains 0.2 to 0.5% agar, just as a semi-solid media. And agar is basically a golden green granular powder prepared from seaweeds, and then it melts at 98 degree. 98 degree it melts, and sets means converted into solid form at 42 degrees Celsius. The liquid media having no agar, available in test tubes, bottle, flask, etc. In semi-solid media, there is 0.2 to 0.5% agar, gel type nature, gelatin like nature, and basically soft, mostly present in the petri dishes and test tubes, and specific for motile and non-motile bacteria separation. Moving towards chemical composition, there are three basic types: natural, synthetic and semi-synthetic. Natural, the first one is the nutrient agar media. Different composition of ingredients were taken. For example, as a carbohydrate source, beef extract was taken. Then for nitrogen source, peptones were taken, electrolytes and solidifying agents were taken and dissolved in 1000 ml or 1 liter water by heating on autoclave at 21 degrees Celsius. Beef broth media is taken at the time. This media is basically used at the time of pasture. Selective culture media of two types: mannitol salt agar, eucin methylene blue agar, for two different species. Mannitol is for Streptococcus species and eucin methylene blue for E. coli species. Different culture medias are taken. In the process of differential culture media, bile salts are taken, specific for gram positive bacteria. Selective is for Streptococci and E. coli, Differential is for gram positive. Example is Agar media. Enriched culture media is basically for the species of Streptococcus and Neisseria. If we want to cultivate Streptococcus species, then we will prepare nutrient blood agar, and if we want to cultivate chocolate agar media using Neisseria species as in Inoculum. Then synthetic media having components of known quantity like glucose, ammonium phosphate, potassium phosphate, magnesium sulfate and sodium chloride are basically used for the purpose of obtaining energy, nitrogen, phosphate and enzyme formation and the internal environment of cytoplasm. Then semi-synthetic culture media, the media which have chemical composition half known. Example most important is PDA, potato dextrose agar. Then special medias were discussed as enriched media, selective media, differential media, transport media, anaerobic media and basal media. Then basal media, very simple media and support non-fastidious bacteria which do not require any nutritional supplement for growth. Then enriched media, specific nutrition is added into it like blood agar and chocolate agar. You can see here blood agar is of red color, chocolate agar is of chocolate color. Then selective media, for specific bacteria different dyes are added, different antibiotics are different inhibitory agents are added for this selective media preparation. Then eosin methylene blue is the basic example. The dimethylene blue in the medium inhibit the growth of positive bacteria. We can see here. Then differential media, for different type of medias are taken, different type of colors are seen. The colony is identified, different colonies are identified on the basis of colors. Different dyes are used for the identification. As in McConkey agar, you can see the color distinguished as pink color for lactic acid bacteria and from colorless for non-lactic acid fermenters. In case of XLE, xylose, lysine, deoxycholate agar, the recovery of Salmonella and Shigella species is taken as. In case of celery agar, cysteine, lactose, electrolyte deficient agar, you can see here the difference. Basically, urine samples were taken for this purpose of study. Then TCBS, thiosulfate citrate bile salt sucrose, is taken as a media for the isolation of basically Vibrioid species like Vibrio cholera or Parahemolyticus species. Further, the transport media basically for transporting the sample from one place to the other place without any destruction. Stewart media and glycerol saline buffer media is basic example of this transport media. Anaerobic media to grow those organisms which are basically anaerobic in nature, which survive in the absence of oxygen. Robertson cooked meat medium and thioglycolate media is a basic example. Now how it is isolated, how we prepare the colonies and how we isolate them. The streak plate technique, the pour plate technique, the enriched culture technique, the serial dilution method, the single cell dilution method and roll tube method. Streak plate we have discussed here. We take a petri dish, put the media there in a controlled environment. We draw different irregular lines. You can see here like 4 different type of colony morphologies distinguished by using this method. These different type of streaking patterns. Then pour plate technique, we simply take 3 test tube with different culture media, with same culture media, with same media, and we put the inoculum taking one loop into A, 2 loops were taken and added in test tube B, then 3 loops were taken from B and added in C. Then the last test tube was examined under microscope after incubating at certain time period. This is all done for culture medias. This is basically the basic introduction of culture media and the techniques we use to isolate that culture we prepared by using the inoculum. That inoculum uses the media, nutrition present in the media, and that is further grown the inoculum species into its increased number. This one also, you can draw this one or this one, both are similar. Just here is original inoculum, 1 ml is taken, then again 1 ml taken, added in the other tube, 1 ml into other, then transferring this sample and taking from the 5th tube and then add it into the petri dish, put it in the incubation for certain time periods at required temperature, and the growth is observed. All these are the methods for isolation. Now when they are isolated, what we have to do? We will have to identify this type of prepared culture. How they are identified? As we discussed earlier, the process of staining, gram staining basically. Then gram staining basically identify either the bacteria are gram positive, either these are gram negative, or either they are acid fast bacteria. That is separate method. Here till now we have discussed media, we have discussed the prepared culture, and then how that culture is grown by using different methods like streaking or like portrait method. After preparing that culture, our next topic will be about the identification of that type of artificially grown microorganism by using basically the staining technique, which identify gram positive and gram negative bacteria. Alright, this is done for now. Thank you so much.